I'm going to be reviewing the Lac Operon, which is a genetic regulatory mechanism proposed by Jacob and Monod, which describes the idea that the control of enzymes in cells is due to transcription regulation in DNA. Lac Operon contains enzymes that allow E. coli to import and metabolize lactose when glucose is low. This is a way in which prokaryotes are able to respond to changes in their environment. An operon is a genetic unit that contains an operator, a promoter, and genes in a single prokaryotic mRNA. The Lac operon contains regulatory genes as well as coding genes. Now, if we go downstream, we start with a repressor promoter. Lac-I, a repressor protein, a cap binding site, a promoter, an operator, Lac-Z, which is beta-galactosidase, Lac-Y, which is lactose permease, Lac-A, which is transacetylase. Now, Lac-Z is an enzyme that cleaves lactose into glucose and galactose. Meanwhile, Lac-Y is a membrane protein that transports lactose into the cell. Now, this single mRNA transcript is polycistronic since it has more than one gene in a single transcript. When glucose is low and lactose is present, CAMP levels end up increasing. The CAMP binds to CAP, which ends up binding to the CAP binding site. This activates the promoter, and so the initiation complex is formed. RNA polymerase ends up binding to the promoter and so allows for the transcription and translation of these three proteins. When lactose is not present, the Lac repressor protein ends up binding to the operator and thus prevents the transcription of Lac genes. This is known as gene repression. Now, when lactose is present, allolactose, which is a product of lactose, ends up acting as an inducer and binds to the Lac repressor protein that is bound to the operator. This releases the Lac repressor protein from the operator, thus allowing for the continuation of the transcription and translation of these Lac genes.